Welcome to the unit on innate immunity. There are two main parts to the immune system, the innate immunity and your adaptive immunity. Throughout this unit, we're going to really focus on the first line of defense, those innate immune system properties that help you combat foreign pathogens as they first get into the body. So we're going to start looking at physical, chemical, and protein defenses that tend to be things that people don't even recognize are part of the immune system. Let's go ahead and take a look. So as we move into this unit, we're really starting the field of immunology. And it's important to recognize that immunology is in and of itself an entire field of study. And so what we're going to cover in this class is really just an overview of the topic. There are entire classes on immunology. We actually have one here at UVU if you want to take it. But there is also entire majors in immunology. And so what we're going to cover over the next two units really is just going to be a surface level about the topic. The field of immunology really focuses on the molecular and the cellular interactions that are happening between the host and foreign molecules that are coming into the host itself. It's the way we figure out what is self, so what are our cells, and how are they different from those foreign molecules, and then how does the body respond to those foreign molecules. And there's two levels to the immune system. So there's what we call the first line defenses or the innate immunity. These are the things that foreign invaders and pathogens will actually come in contact with first when they get into the body. And then there's the second line, or the adaptive immunity, that we'll talk about in the next unit. So the defining feature of innate immunity is that it's non-specific. So when we talk about the concept of immunity, you often think about that idea of now being immune to something. So if you see it again, you're not going to get it. That's not part of the innate immune system. So the innate immune system is able to recognize that something is foreign, but not what that foreign thing is. So it's the difference of saying you're not self versus you're E. coli or you're influenza. Right? When we say non-specific, that's all we mean. We know it's not self, but we don't really know what it is. And because of that, it doesn't confer that memory or that immunity. If you don't actually know what something is, there's no way you can develop cellular memory to it. That's going to happen with the adaptive system that we'll talk about later. There's two parts to your innate immunity. There are what we call the non-cellular defenses. These are the very first part of the innate immune system, and they're often things that people don't even think about as being part of an immune system. So there's physical things like barriers or your temperature. There are chemical things like enzymes or just chemicals like salt or hydrochloric acid, low pH. We have fevers, and then we have different proteins that are involved in these defenses. There's also cellular parts of the innate immune system, and so we'll talk about the different cells that make up the innate immunity and what their job is, what their role in phagocytosis is, and we sum it all up in the end, which is inflammation, which really brings all of these innate immune properties together into one thing. The physical immune defenses are things that physically stop microbes from actually even getting into the body. And so they are things that are barriers. Physical barriers like your skin. Your skin is actually your largest immune organ. So nothing can pass through your skin unless you have a cut, okay, unless there's a break in that barrier. And also your mucous membranes. So all along the respiratory tract, all along your GI tract, you have mucus layers that are there to help trap pathogens so that then you can mechanically get rid of them. And that's the other physical defense, is the mechanical removal of microbes. There are things like sweat that washes from your skin or tears. It's why if you get something in your eye waters, it's that flushing of the system to mechanically remove those microbes. This is also why the symptoms of a lot of diseases are mechanical removal. Things like vomiting or diarrhea, if you have a gastrointestinal issue, is a way to get those microbes out of the body. Other mechanical ways are your mucociliary escalator. So in the respiratory tract, it's those cilia that beat up, again, to stop pathogens from coming in, or things like when you cough or you sneeze. Those are all mechanical mechanisms to get those microbes out of the body before they can cross those barriers and get into the bloodstream or get into the tissues where they can actually cause infection. Along with the physical barriers, there are also chemical defenses of the innate immune system. And these are chemicals that will actually kill cells. So whether they're chemicals like inorganic molecules, such as the salt on your skin that makes for a saltier environment so microbes can't live there as easily, or the hydrochloric acid in your stomach. A lot of things you consume have microbes on them, but they get destroyed in the stomach acid, so they don't actually travel to the rest of the body. or whether those chemicals are organic molecules like enzymes that are actually functioning to cleave and degrade and fight off these pathogens. So things like lysozyme. So lysozyme will cleave the peptidoglycan cell wall. You cleave that, remember they're going to lyse from osmotic pressure. And so lysozyme is found in things like tears, again, protect your eyes. It's found in your mucus layers, so we can trap them and then we can destroy microbes. And then your cells also produce a whole lot of antimicrobial peptides. And these are just proteins that have some sort of activity, so they're enzymes that will fight off pathogens, typically called things like defensins or bacteriocins. They're enzymes that are going to damage cell membranes or DNA or cell wall, some properties of those bacterial cells. And so these chemical defenses work in combination with your physical defenses to both stop microbes from coming in and to try to kill them before they get into the tissues of the body. Interestingly, fever is not something we typically think of as associated with the immune system, but it is actually a defense mechanism. So a fever is defined when your oral temperature is above 38 degrees Celsius or 100.4 degrees Fahrenheit. And it's caused by something called pyrogens. So there are some microbes that actually produce molecules that act as pyrogens. Those pyrogens set off our body processes that will communicate with the hypothalamus and control that thermoregulatory center. And so the body temperature increases in response to these pathogens being in the body. The purpose of that is that elevated body temperature is going to reduce bacterial growth. So remember when we talked about temperature ranges and classifications, your mesophiles, those organisms that are typically going to be your pathogens, their optimum growth temperature is 37 degrees Celsius, which is your body temperature, your standard 98.6 degrees Fahrenheit. When we get above that optimal temperature, bacterial growth slows down because we're no longer at that optimal temperature. And so we can see here in the graph, the blue lines are your body temperature. The red lines are your bacterial growth rate, so how fast are the bacteria growing. As we start to get a fever, so here we're at 100 degrees, so we're just running a fever, and you'll notice the bacterial growth has decreased to 86% of its rate, so it's no longer growing at 100%. If we increase that temperature even more to 103 degrees, now we've decreased the bacterial growth, all the way down to 55%. Okay. The higher the body temperature, the slower those bacteria are growing, and the slower they're growing, the less chance they have of spreading through the body. Now, here's a problem. We've got this dotted line here in the middle. Everyone recognizes that as the body temperature increases, it will eventually get too hot even for your own cells to function. This is when we start damaging brain cells. Can lead to things like seizures. And so this is why once you hit that kind of 104 range, you really need to work on getting that body temperature down so that you're not harming your own self cells. But notice if we get all the way up to 104.5, our bacterial growth is way down to 36%. Unfortunately, we are also now in the harmful fever side of things. I point this out because the entire immune system is like this. You're always going to be walking this fine line in immunology where we need to have enough of a response to stop the pathogen, but too much response will harm our own cells. And so your immune system that's destroying foreign invaders can also damage your own tissue quite easily. So regulation of the immune system is just as important as activation of it, because you don't want all those immune processes just running unchecked. You'll end up with things like this where the body temperature just keeps going up, and that's a problem. But also remember that this fever is actually beneficial. So realistically, if you are sick and you're running a fever of 101 and you can deal with the fever and the chills that kind of go along with having a fever, you will have a shorter illness if you just ride it out. When you take medicine to decrease the fever, you're also losing this effect of the innate immune system. Now we're going to look at some of the protein defenses of the innate immune system, and the most important are going to be complement proteins. So complement is a whole series of proteins, over 20 proteins, that are circulating in the bloodstream. And they're circulating in a conformation that is off. So the complement proteins do not function all the time. They only want to function if there's some sort of foreign molecule that has gotten into that bloodstream that now we need to combat. Okay, so the complement proteins can lead to pathogen lysis. Okay. They can also help recruit immune system cells, so to say, hey, there's a problem over here, we need to come fight it. And they do something called opsonization, which we'll talk about a little bit later. It really just helps your immune cells function better. As we're looking at complement, remember its main purpose is to prevent against bloodstream infections. So those proteins are part of your serum proteins. They're there all the time, ready to go into action. But they will also diffuse into tissues during inflammation. So they can fight pathogens out in the tissue also, even though they're circulating and they reside in the bloodstream. There are 3 ways you can activate this complement proteins. So remember, they're inactive, they're floating around in conformations that aren't functioning. We need to turn them on and to activate the pathway in order to get them to work. Those 3 ways are called the classical pathway, and this involves the bonding of antibody to a surface. The alternative pathway, which actually happens directly by the bacterial cell itself. There are proteins on the bacterial cell that will activate complement protein. And then what we call the lectin pathway, which is activated by lectin that can also be bound to a bacterial cell. When we go through complement, you guys don't need to know how the actual activation happens, but all of these pathways come together at a complement protein called C3, and that's where we're going to start the pathway from. So on the next slide, I'm going to walk you through how complement is activated. It kind of functions like a series of dominoes, where one protein activates the next one, which activates the next one, and so on and so forth. So remember, there's 3 ways we can activate the complement pathway. There's the classical, which is going to be activated by antibodies, these little Y-shaped proteins. There's the alternative, which is going to be activated by the bacterial themselves. And then there's lectin, which is going to be activated by lectin molecules bound to the surface of the bacterial cell. But no matter how we activate, and there's different complement proteins involved in each one of the activation pathways that you don't need to know, when we activate to those, we're all going to converge on a complement protein called C3. And so we're going to walk through what happens once C3 becomes activated. Down here at the bottom, this is going to be our bacterial cell. And this is where all the complement pathways are going to be converging to. Okay, so we've activated complement. That activated complement is going to come down and say, okay, C3 is the inactive form. When it gets activated, what happens is that C3 gets cleaved, so we actually break a piece off into what we call C3A and C3B. Okay, we've broken this little piece off. C3B binds to the surface of the bacterial cell. C3A diffuses away into the body, and it's going to recruit other immune cells. So those immune cells are going to do something called chemotaxis. And they are following a chemical gradient, so the C3A back to where it came from, so that we can find where the infection is. What that means is that C3A is what we call a chemotactic agent. Okay? It induces chemotaxis of cells. So C3B is now our next complement protein. It is bound to the bacterial cell, and what it's going to do is it's going to take C5, another complement protein that's just circulating in the bloodstream, and C3b can now cleave C5. So again, we make a C5a and a C5b, which also binds the bacterial cell. C5a diffuses away, and it can also recruit those immune cells. C5b is now activated, and it's going to recruit to the cell surface a bunch of other complement proteins. And these ones aren't cleaved, they're just now able to bind because C5b says, hey, come over here, bind here, this is a bacterial cell. So we bind complement proteins C6, C7, and C8. Now, once 5b, 6, 7, and 8 are all bound to the surface of the bacterial cell, all of them working together can recruit C9. And C9 is where all the action happens. So C9 actually recruits multiple subunits, and they form this pore in the cell. So they actually punch a hole right through the cell, and what happens is all of the cellular contents from inside now spill out, and that's going to lead to lysis of the cell. So you can think of C9 as physically poking holes in the cell membrane, in the cell wall of the bacterial cells, leading to lysis of the cell, which is why this entire complex, so your C5b, 6, 7, 8, and then all of your cruded 9, is called the MAC, or the membrane attack complex, because this is what's physically poking holes in that membrane, which leads to lysis of the cell. Remember, complement pathways themselves can lyse the bacterial cells so they can directly kill them. They can also recruit immune cells so they can help bring other immune cells into the area to fight these off. And anything that has C3b bound to it is what we call opsonized. Okay, that C3b can also act as a flag for our immune cells that say, hey, come destroy this bacterial cell. Okay, so 3 things that complement's involved in: your lysis, your recruitment, and your opsonization. The other proteins of the immune system are involved in both the innate defenses, but also the adaptive defenses. So the cytokines are these immunomodulating proteins. So they're proteins that help control the immune system. And they're really important for cell signaling. And so you'll see a lot as we go through the processes of immunology that this involves cell communication. It's all about this thing activating this next cell, which then does this process, which is going to activate another cell, which does this process. That activation, that communication is controlled by cytokines. So you can kind of think of them as the voices of the immune system. They're going to be produced by both innate and adaptive cells, and their effects are what we call pleiotropic. So what that means is that one type of cytokine might have one effect and communicate one signal if it's interacting with a certain cell type, but have a completely different effect if it's interacting with another cell type. Because of this, cytokines can get quite complicated, and we're not going to go into the individual roles of each individual type of cytokine throughout this kind of general overview. I more want you to understand that these things can act both locally and systemically to communicate immune responses, okay, so that cells can talk to each other. There are 4 main classifications of your cytokines. They are chemokines, which are things that induce chemotaxis. So again, those chemoattractants, the molecules that cells can follow. There are interferons, which are going to interfere with viral replication. And we are going to talk about them briefly on the next slide because they're one of the main ways we deal with viruses in the immune system. There are interleukins, which are going to really be involved in your adaptive system to help with T cells and B cells and their development. And then there are things that are called tumor necrosis factors that actually help promote apoptosis of our own cells when there's something going wrong with our own cells. So your cells can actually become foreign to self. Okay, that's really what cancer cells are. They've picked up mutations and they're no longer functioning the way self cells should. The immune system has ways to try to handle those cells also. So whether the foreign molecules are coming from outside the body or whether there's something wrong inside the body, we can still recognize that they are not self anymore and try to handle those. So interferons are the cytokine classification that's really involved with trying to stop viral replication in our cells. You can think of them, their name coming from they interfere with viral replication. Interferons are proteins that are able to be produced by just about all of your body cells because all of your body cells can actually be infected with a virus at some point. So you can see this cell here has been infected with a virus. And in response, so the virus is undergoing those viral replication steps that we talked about. And while it's doing that, the cell has turned on its production of interferon. This is an example of gene regulation. Okay, so the gene in the cell is now being transcribed and translated. So the cell makes interferon and it secretes it out into its environment. This works locally, okay, to bind to the neighboring cells. Okay, you can kind of think of this as the cell taking one for the team because this cell here is going to be killed by the virus. Okay, so this cell is going to die due to that viral infection. But before it does, what it's done is it sent out this warning signal to the neighboring cells. And so that warning signal is telling these cells to get ready to destroy RNA and stop doing protein synthesis. What that's going to do is that when this virus comes over and tries to infect this cell, it's going to get in here. Remember, it's going to release its genome, and then no transcription or translation will happen. If no transcription or translation happens, the virus can't replicate. The virus doesn't replicate, it doesn't spread through the tissues. It's also going to signal cells that might be infected. So if there's any infected cells around it, it's telling them to undergo apoptosis. So notice this virus has gotten in, but we haven't started making lots of other viruses yet. If this cell undergoes apoptosis with its controlled cell death, it's when the cell actually says, okay, there's something wrong, and it's going to remove itself from the population. That cell dies, this virus can't replicate, we don't spread through the immune system. And then the last thing that it will also do is it will activate immune cells to come into the area and help combat the virus itself. But these first two are really the most important. If that virus leaves this virally infected cell and goes to infect neighboring cells, but it can't replicate there, the virus can't spread through the tissue, and we've really stopped the viral infection in its tracks. So I always like to end with this slide to show you guys that this is what I mean when I say cytokines are quite complex. So each one of those little letter number combinations, like IL-3 stands for interleukin 3, is a separate cytokine. And if you notice that TH cell in the middle is producing IL-3 that's going towards the eosinophils, it's producing IL-3 that's going towards the mast cells, it's producing IL-3 that's going towards that CD4 cell. This is interacting with lots of different cell types, and it's doing something different in each one. So you'll see there's lots of arrows going around. There's lots of names of cytokines. These are all the cell communication that has to happen so that your immune system functions when it's supposed to, does what it's supposed to, and then shuts itself off when the problem is no longer there. We need to be able to communicate with cells so that they activate and go do their job, but then also so that they deactivate and stop doing their job so that we combat foreign molecules, but we don't go overboard and go against self and start homing your own tissues too. And that's really the important role of cytokines, is to control that cellular communication throughout the immune system.